Hi everyone, welcome to NEET Biology channel. A botanist removes the integuments from a developing ovule. Which key process during fertilization would be directly impaired? A. Formation of embryo sac. B. Pollen tube entry through micropyle. C. Mitosis in the functional megaspore. D. Development of funicle. Correct answer is B. Pollen tube entry through micropyle. Explanation. The integuments of an ovule form a small opening at one end known as the micropyle, which is the primary route through which the pollen tube enters to deliver sperm cells during fertilization. If the integuments are removed, this entry point disappears, directly impairing pollen tube guidance and entry. While the embryo sac forms inside the nucellus and not the integuments, and mitosis in the functional megaspore is an internal process unaffected by the presence of integuments, the funicle is a stalk-like structure that connects the ovule to the placenta and is unrelated to micropyle formation. Therefore, the most directly impaired process is pollen tube entry through the micropyle. During a genetic study, a plant shows multiple embryo sacs in a single ovule. What condition does this represent? A. Asexual reproduction. B. Sterility. C. Polyembryony. D. Gynoecium fusion. Correct answer is C. Polyembryony. Explanation. Polyembryony refers to the development of more than one embryo within a single ovule, often due to the formation of multiple embryo sacs. This is a deviation from the norm, where a single functional megaspore gives rise to one embryo sac. Asexual reproduction, such as apomixis, might also produce multiple embryos, but the presence of multiple embryo sacs specifically defines polyembryony. Sterility would imply failure of reproduction rather than excess development, and gynoecium fusion pertains to the structure of the carpels, not embryo sac formation. Hence, the correct condition represented is polyembryony. In an experimental plant, the Nucellus degenerates prematurely. Which structure would fail to develop as a direct result? A. Integuments. B. Micropyle. C. Embryo sac. D. Funicle. Correct answer is C. Embryo sac. Explanation. The nucellus provides nourishment and physical support to the developing megaspore mother cell, which undergoes meiosis to form the functional megaspore that later gives rise to the embryo sac. If the nucellus degenerates prematurely, this entire sequence is disrupted, leading to the failure of embryo sac development. Integuments, micropyle, and funicle are separate structures. They are not directly dependent on the nucellus for their formation. Therefore, the structure most directly impacted is the embryo sac. A mutation blocks mitotic divisions in the functional megaspore. What would be observed in the ovule? A. A mature embryo sac. B. An 8-nucleate structure. C. Only a single nucleus megaspore. D. A fertilized egg. Correct answer is C. Only a single nucleus megaspore. Explanation. Normally, the functional megaspore undergoes 3 mitotic divisions to produce an 8-nucleate embryo sac. If a mutation blocks these mitoses, the megaspore will remain a single nucleus structure and will not progress to form the organized embryo sac. Options A and B refer to the fully developed form of the embryo sac, which is impossible under these circumstances. A fertilized egg, option D, cannot form unless the embryo sac has been completed, so the only observable feature would be a single nucleus megaspore. Which part of the ovule should be targeted to prevent pollen tube guidance during a gene knockdown experiment? A. Egg cell. B. Synergids, filiform apparatus. C. Antipodals. D. Chalaja. Correct answer is B. Synergids, filiform apparatus. Explanation: the synergids, particularly their filiform apparatus, are specialized cells adjacent to the egg cell that release chemical attractants guiding the pollen tube to the correct location for fertilization. Disrupting gene function in these cells would impair pollen tube guidance. The egg cell is involved in fertilization but does not guide the pollen tube. Antipodals and the chalaja are not involved in directing pollen tube movement. Thus, targeting synergids is the most effective way to interfere with pollen tube guidance. A plant ovary has only one ovule. What is a likely candidate species? A. Watermelon. B. Mango. C. Papaya. D. Orchid. Correct answer is B. Mango. Explanation: among the listed options, mango typically exhibits a single ovule per ovary. This is a distinctive trait also shared by wheat and paddy. Watermelon, papaya, and orchid generally have multiple ovules per ovary. Therefore, if a plant displays a single ovule within its ovary, mango is the most likely species among those given. If a flower has free carpels, not fused, which condition is it showing? A. Monocarpellary. B. Syncarpous. C. Apocarpous. D. Bicarpellary. Correct answer is C. Apocarpous. Explanation: an Apocarpous gynoecium consists of multiple free, unfused, carpels, each forming a separate pistil. In contrast, a syncarpous condition features fused carpels. Monocarpellary means only one carpel is present, and bicarpellary refers to two carpels, which may or may not be fused. The question emphasizes the free nature of the carpels, making Apocarpous the correct term. Which technique would best reveal the number of nuclei in a developing embryo sac during megagametogenesis? A. Gene sequencing. B. Nuclear staining with microscopy. C. RNA blotting. D. Dissection under UV light. Correct answer is B. Nuclear staining with microscopy. Explanation: nuclear staining followed by microscopy allows direct visualization of nuclear divisions during megagametogenesis. It can effectively track the transition from 2 to 4 to 8 nucleate stages. Gene sequencing and RNA blotting provide genetic information, but do not visually reveal nuclear numbers. Dissection under UV light might help with structural observations, but it doesn't provide clarity about nuclear division stages. Therefore, nuclear staining and microscopy is the most accurate method for this purpose. A plant lacks polar nuclei in the central cell. What outcome is expected after fertilization? A. A triploid endosperm. B. No endosperm formation. C. 2 embryos. D. Micropyle formation. Correct answer is B. No endosperm formation. Explanation: in angiosperms, the formation of the endosperm requires the fusion of one male gamete with the 2 polar nuclei in the central cell to form a triploid structure. If the polar nuclei are absent, this fusion cannot occur, resulting in the failure of endosperm development. While option A describes normal conditions, it is impossible here. Option C is unrelated to central cell activity and more associated with polyembryony. Micropyle formation, option D, occurs earlier and is unrelated to the central cell. Thus, the correct consequence is the absence of endosperm formation. During pollination studies, a researcher aims to trace the pollen tube's path. Which part of the ovule should be tagged with a fluorescent dye? A. Chalaja. B. Antipodals. C. Micropyle region. D. Nucellus. Correct answer is C. Micropyle region. Explanation: the micropyle is the gateway through which the pollen tube enters the ovule to deliver sperm cells. To study the path of the pollen tube, tagging this entry region is essential. Chalaja is the basal part of the ovule and is not involved in entry. Antipodals are located at the chalajal end and do not interact with the pollen tube. The nucellus surrounds the embryo sac but is not the first point of entry. Therefore, the micropyle region is the correct target for fluorescent tagging.